Good morning.
So, is that everyone now?
I think, no, Yomali, also Yomali is still missing.
I don't see anyone connected with that name. Alessandro, Cristina, Javier, Jose.
Oh, I think that she's changing her name to the UTEC Yomali. So she's going to enter again.
Okay, so I believe we can start the questions now. You will have 15 minutes to ask and answer questions. So I'm gonna let some of the judges here that are joining us. If you're gonna briefly introduce yourselves, that would be great. Thanks.
Thank you so much and welcome to the team. Really nice project, and we really enjoyed going through their Juggle page and also the PDFs. My name is Mohammed Tarek. I am a medical intern and a research associate in bioinformatics and synthetic biology. My work mainly focuses on immunotherapy, so I really enjoyed this project. Thank you.
Hello, everyone. Good morning. My name is Carolina Bonilla. I'm from Mexico, and I have some experience in molecular diagnostics and COVID, so your project is really interesting.
Great, so feel free to continue asking any questions that you have on the students project.
Thank you so much. So I'd like to start with to congratulate you on your project, because really it seems that you have put so much science and also dedication into it. You guys are all undergrads?
Yes.
Great.
Yes, we are.
Okay. So are you from different majors?
We are 13 from bioengineer.
Yes,
One is
great.
from electronic engineer, and one is from computer science.
Wow. Okay. So I will start like by specific points that we can go through, and just remember that one of the main keys for these questions and for this session is not just for the assessment, but also to improve your project, which is really great. So the first point is that you have mentioned the selection for the epitope prediction, different tools, and you have mentioned something on the cytokine predictions, but we can't see any tools used for specific cytokines release upon a simulation by your epitopes. So have you performed such simulations, or is it in your future plan?
Yes, we have performed a couple of simulations that were regarding to the modulation and validation of the three-dimensional structure, the immunogenicity, antigenicity, and also the thermostability.
Okay, great. So I think you can just, of course, to predict the response of cytokines upon recognition of your epitope, this is really important. So you can use, there are some tools that you can use, or even you can develop your own models that can, help you to set of like predicting the interferon gamma response for your epitopes and so on. So the second question is that you have mentioned your approach for using the subunit of the receptor binding domain. That's one example for COVID. So do you think that you need to, for example, if COVID releases some evolutionary mutants or some variants like we see, do you think that your approach is ready to tackle these variants, which could make your subunit vaccine susceptible to escape and reduce its effectiveness?
Okay. Well, receptor binding domain is you say it's a region located in subunit 1 of SARS-CoV-2 spike protein. It interacts with human ACE2 receptor. We found that receptor binding domain in vaccines against SARS-CoV-2 was be able to generate protective antibodies which neutralize the entry of virus into human cells. Also, it has been reported that neutralizing antibodies against this region can be remain in different strengths. So we think that this domain is a good candidate to be added in the vaccine.
Yeah, thank you so much. Please, Dr. Carolina, you can go on. If we have time, you can resume with some questions.
Thank you, Mohammed. Well, first, I think that this is a really good project because you are undergraduate and you think in all the different things that you need to think to become a real project that could be implemented. So this is impressive. Congratulations. I have some questions about the other part of the project that you think about it. About the technique of purification of the vaccine. Do you have some technique for the future of this project?
Just so you know, we are also allowing questions to be asked in Spanish, or Portuguese, but the answer needs to be in English. So.
Yeah, so right now the project and the scope of the project, as we already we have explained in the job, is to prove the thermostability of this protein. So we know that a vaccine is has a really complicated process. It's not simply like producing the protein and then you can put it on people. So we also have in consideration the clinical trials that have to put through the vaccine. So we have already talked with some pharmaceuticals, and that's also a question that they always ask. We are going to first prove this important part, the thermostability, and also have another essay that is the ELISA. It's more with the immunogenicity part of the protein. But after this, what we are planning to do is with a pharmaceutical that could be interested. For example, we have with Roche kind of collaboration because they have the capacity of doing this kind of clinical trial that need a lot of logistics. But right now we're focusing on this part, really important with the experimental part to prove that it could support these high temperatures. And then we also have like some plans about how we could put this product in the market.
Okay, good. Did you run some in silico analysis to evaluate the efficiency of the vaccine compared with the others in the market? How do you can pass for this design step and go to the other part of implement? I mean, if you run the thermostability test and the other test, how can you be secure that this is the good project, well, not the good project, the good vaccine to scale?
Yes, for that, we have developed a series of bioinformatic analysis in order to prove in silico that thermostability first, of our chimeric antigen, we have choose different softwares that allow us to compare how will behave the receptor binding domain alone and how will behave with the adjuvant, in this case, the lectin rising toxin B. With that, we have found in our results that the thermostability of the antigen is better than when it is alone. We also have the support of the scientific evidence of this carrier to improve thermostability in other antigens. And well, that's what was our analysis in silico. We also have made an analysis for the immunogenicity validation that was with different software also to know how which antibodies and so on does the receptor binding domain induced in our analysis, and with that lectin also a joint to this. All these analyses are having made in order to prove the concept that we are looking for to prove in the experimental part of our project.
Okay, thank you.
Yeah, I think that was a very important question because it has really big potential to improve your project, because really the type of analyses that you have done for specific epitopes are actually to detect the immunogenicity on a specific segments of the receptor binding domain. It actually does not reflect how the immune populations of cells or cytokines or antibodies would be released within patients in a clinical trial, sorry. So I think one type of analysis could be, or have you considered just to simulate the immune response in a patient? There are some computational models that can do this. Do you think that this would help your project in the near future?
Of course, yes. We continue working in the project. We will not stop here. We will prove and use another softwares like the one you have mentioned now to demonstrate and validate more the concept that we are stating in the project. But yes, it's important to consider other softwares, other programs, and bioinformatic tools to support our idea. To have, yes, that's important.
Yes, great, thank you. So
thank you for your recommendation, by the way.
Thank you. I wish you the best of luck on it, because I really liked also the point of thermostability and how are you putting it into a local context and how this would solve the problem, not just in your nation, but also in other nations in which the thermostability is an issue for vaccine delivery and the cold chain. So have you thought about like maintaining a version of your vaccine that is population specific, in which you know the HLA alleles that represent your population, or the population of your nation, and also you can reproduce the design for other nations of different allele phenotypic construction? Have you considered this into your project?
We haven't not considered that, but for example, our vaccine is very same like a BioNTech vaccine, and we have performed a microarray assay with with the gene expression of vaccinated patients of different nations with the BioNTech vaccine, and they haven't had like different representations of phenotypes or efficiency of the vaccine. So I suppose that is the same, and with the addition of the adjuvant, that will improve the immune response.
Okay, thank you. So you are rather working on a general scale for one vaccine that could fit most of the population. It's really a nice strategy, but also just make sure that it can also fit with some escape variants that the virus could develop, or some populations that could have different immune phenotypes. So I really liked also the part in your collaboration in which you have received feedback from other experts on the flexibility analysis for the thermostability and they have mentioned to use some molecular dynamics simulation software like GROMAX, I think. Of course, yes, one of the limitations of molecular dynamics simulation is how costly it is to perform on regular machines. So I think you can, one of the methods that you can try is to try collab the molecular dynamics, like using NAMD or other molecular dynamics software. So you can run it on a shared server, like on the cloud, like on Google Colab. Do you think this would help you? Because the flexibility, actually, the thermostability actually depends a lot on the flexibility of your design. So do you think that this would help you better estimate? I know that you use a scope and other physicochemical estimations, but do you think this would be helpful?
Yes, it will help a lot because, in fact, I was doing like the simulation with GROMAX, but only I do the RBD, and it goes like 15 hours at different temperatures, and I couldn't do the I couldn't do with the RBD plus the RTB, and I didn't know why until we made a collaboration, and a guy told me that GROMAX has like a limit of the size of the structure, and then I have to use like a corset grain, and for that reason we only present the result of the scopy, because in fact in GROMAX I couldn't do with the composite protein.
Great, thank you so much. So
sorry, I will need to close this section now. So thanks to the judges, thanks to the team. So please vacate the room, and I'm going to start letting the next team. And congratulations on your judging session.
Thank you so much.
Thank you very much.
Thank you.
Thank you. Bye.
Thank you so much for your time.
Thank you.
Hi there, team. So yeah, I have that you are the only team members with the correct name. Is anyone else missing? And it would be great if we can do this as soon as possible.
Yeah, we're missing several teammates, but I think that they are just changing their name.
Okay. So starting now, you will have 18 minutes, just so you know.
Okay, I think we're missing like half of the team.
Okay, so you can start telling me their names.
Ana Laura, Mariana, Muñiz, María Fernández Queda, María José Rojas, Kevin, Oli, Emilio.
I don't see any Emilio.
Ya no.
I see Álvaro, Aura Tamés, Carlota.
Álvaro, Aura. Yeah.
Cristina, Javier, José Mario.
No, Mario. Jimena.
Yeah, I don't see Jimena.
No. Monse. Monse.
Fernanda?
Fernanda.
No, so I think we can start with the questions. We can jump straight to questions, and if I see anyone else with the CCM team, I'll let them in. So welcome to your judging sessions. You now have 16 minutes.
hello, team BioTec-CCM. Thank you for your very nice project. I really enjoyed it, and I would like to congratulate you on the amount of effort and scientific determinants that you have put into that project. I really enjoyed it. I would just have some quick points to discuss with you, and be really sure that part of these points is just to help improve the project further, which is really great at the moment. So I would like to start with the toy hold structure. So do you think that you have some kind of computational characterization for the dynamics of the toehold binding, or how this structure would fit into your design?
Do you mean that if we modeled this interaction of our toehold and the binding of the aptamer?
Yeah, exactly. And also on the expression of a stable variant of the toehold, transcription. Yes, so the toehold itself and also the interaction.
Sorry, I didn't get your last state.
Sorry, so I meant that also, yes, the interaction, but also how do you ensure that the sequence of the toehold is actually releasing a structure that fits into your design? So basically, you can do it, yes, but like you have mentioned, by modeling the structure itself rather than in an interactive state.
Okay, so we modeled the interactions. It is all very well specified in our mathematical model supplementary file. But if you mean about if we actually design like the toehold switch system, we took some bio parts that were already standardized for iGEM, and the only thing we did was to fuse them to our aptamer, so we can recognize our proteins. Well, the proteins were in order to then express GFP in the toehold switch system. Do you need any more details?
No, great. That's great for 1 point. Do you think the interaction would need some insights into its thermodynamics, or are you happy with that, the current state?
No, of course, we will need to improve our model, our design. We are pretty sure about the assumptions we made, where we try to make the model very representative of what we are trying to design. But we know that in the future we will need to experimentally determine some of the constants, some of the interactions that we are not considering right now. But yeah.
Thank you so much. So if you have gone into a state in which you are studying the interaction, what type of interaction would you like to consider? The aptamer interaction or the toehold interaction?
Well, for the model, we actually considered both of them, the interaction between the aptamer with each biomarker, and also the interaction between the toehold with the trigger to express the GFP protein.
Okay, so have you performed it, because I haven't met that paragraph to your PDF? Have you performed some kind of docking of the aptamer into the target protein?
We didn't get to that point because we were having some struggle identifying, with the software and all the bioinformatics. We definitely considered that it was important in order to determine the interaction of the aptamer with our target molecules. And we know some softwares that we could use in the future once we have more experience regards bioinformatics. But for this model, we used the concept that were reported on the literature of the aptamers that we found, because in order to find these sequences, you have to do an experimental design. So that's why we use the ones that were reported on the literature, but definitely we would like to do some bioinformatics to know specifically these interactions between our target molecules and our aptamers.
Yes, great, great. This would really help your project further. And also you can consider like type of RNA protein binding or something, but really it's cool right now. So that brings me to the point of aptamer selection and optimization. You have mentioned CLX as one of the approaches that you are going to experimentally use for your proposed validation. So do you think that you can computationally interfere with the process, because you know CLX receives a big amount of lab resources into it in order to optimize for a specific fit aptamers? Do you think that you can computationally interfere in order to make this process a lot easier, a lot less resource intensive?
Yes, well, if I may add, we also identify some specific software that we will use in the future. One of them is Aptani, where you can reduce your CLX, well, the number of cycles used in the CLX process by sequencing your sequences and then introducing them into this software, so they can give a first approach regarding the interaction between the aptamer and your target molecule, so you can have a better pool for your second CLX process. And this drastically reduces the number of cycles. Instead of 60, that is one of the average that it is used in this process, you can reduce it with this software. One of them is Aptani, and the other that we researched a little bit is Aptasut. So yeah, we definitely consider introducing bioinformatics to improve these wet lab experiments.
Great, that's really impressive. So, so you actually depend on GFP for your characterization. So I have gone through on your modeling simulations. And actually, what is really I need some like insights into it, because you considered a directly linear relationship between your input target protein and GFP expression. So do you think that this relationship really mimics what could you meet in a real situation in which you are diagnostically performing your test on real human samples? Because you know there are a lot of impurities, a lot of even for a cell-free system, this could be a lot of biomolecules that render the chemical reaction rather imperfect. So do you think that your modeling could fit into this, like a stochastic process of impurities or something?
Yeah, we need to like take into account all of these things that are not pure. I think we've got today's graph with these graphs because we made a lot of assumptions. So it was like I know it's not completely a linear graph. I think it's a radical modeling, but in the range that we are analyzing our concentrations, it just turned to be linear. So yeah, I think that with taking into consideration all the things that we assumed, like the real values and the real interaction that are happening, we think that there will be a different, it will have a different approach and it will have a different behavior. But I think that this model was able to predict in some way what we're expecting into the wet lab experiments.
Thank you so much. This is really a very beautiful answer. So I think I'm pretty happy with your scientific efforts. I would like just finally to know if you have in your cell-free system, like I'm targeting some protein that is naturally expressed in a different system. Do you think you need to code and optimize this target for your system, or you think there is no need for that? I mean, to do codon optimization such that the frequency of the expression is rather fitting into your cell-free system?
I think there was a little bit of background noise, so we couldn't understand everything. Can you repeat the question, please?
Okay, I'm sorry. I'm just saying, if you are using some coding sequence into your system in the cell-free environment, do you think you need to code and optimize that sequence in order to render your process perfect enough, or you are happy with the coding frequencies used for that system?
Now, as we said before, I think all the sequences were retrieved from the literature. So we actually need to get the ones that are optimal for our models. And I think we just need to get them, or get them from the wet lab experiments to see what's the best sequence that fits in our models, so we can optimize from our results.
Thank you so much. I'm really impressed by your answers and your project. And finally, I would like to have some insights from you on how do you see your project as in a bio-entrepreneurial journey? Do you think that your diagnostic approach could compete with some even national or international startups or big bio-entrepreneurs that are working in the same field?
We see that point. We see that some other business have some kits of this kind of biomarkers or proteins, but we see that these are only focused on the researcher, and we try to focus more on the diagnosis and therapeutic test. And we see that it's possible to have like this startup and this business strategy to develop our test as an entrepreneurship. That's we focus on 4 stages, and these 4 stages are like the resources or the important things that we need to develop our product.
So are you considering applying to accelerators or incubators in the near future?
Can you repeat me the question, please?
Sorry, I mean, are you considering applying your startup into some accelerator program or an incubator program in order to start your journey? Or
we see some platforms of crowdfunding for the first step of our project, and we see that these platforms are beginning to increase the development of other scientific projects like Explore.com, that this is a new platform that start 180 or 780 projects that are good and that develop their projects, scientific projects for this way. For the first stage, we consider crowdfunding. For the second stage, we consider private and public invest. And this could depend on the stage that we have. For the testing and the development of our prototype, we consider only crowdfunding. But for the rest, we are trying to consider other ways of financial.
thank you so much. This is really impressive. So I think you have a bright entrepreneurial future if you really continue your hard work on this as well. And also, I would like to know, how do you consider your diagnostic approach as a product? Are there some, have you considered some estimations for the price? Do you think that the general public could accept it, or this should be integrated into medical insurance in order to render it a product for the public? Have your integrated practices help you to focus on that?
We see that the population is interested in our product. With the focus group, we see that we have 3 principal stakeholders, and we detect that our focus group is interested in these topics, and well, we have a second stakeholder that was researchers from the Mexican National Institute of Geriatrics, and they show us that this topic is important. And we develop our test as a product. We don't have the opportunity or the time to consider the cost of the product, but obviously for that, we consider a table of the principal age-related diseases that are impact our population or Latin America population, that are like diabetes, Alzheimer, and all those things that increase the cost of the population that is aging and that have more than 60 years, and for 2050 will be increased. And we saw that our product will impact that. We don't consider the cost of our prototype or our final product because we don't have time, but we see the cost of the diseases that we have. And these tests, or we consider our product as a test that in a long run could impact and could have like the way of an app that could have a diagnosis and a contact with some doctors. And I think that it's more like that.
And if I may add something, I would like to say that we also consider that its application could be from the medical sector, maybe to have some, or to develop more confidence to the community. So if the doctor or the healthcare could tell them about this test and begin with an in situ application, then they could develop more confidence so they can later try them at home themselves and be more self-improved and feel more confident about it. And also, because we propose a community lab test, we would also like to apply it in medical sector, in clinics, in that kind of spaces where people feel confident and safe, and they know that this product will be not a lie, because we saw a lot of misinformation regarding new technologies and new products. So this way we could make the community more comfortable and more safe regarding these new tools. But yeah, and also like the last strategy will be to distribute it in like pharmaceutical companies, so that it could be available and accessible for everybody. So yeah, we have to consider different sectors for the distribution of our product.
Thank you so much. Very insightful answer. So finally, I really enjoyed the discussion with you. It seems that you have very good preparation for your session and also for your project. And finally, I would like, Jose, if we have time, to know from you, how was the experience? Because this program, this design league, is a really new program. And have you enjoyed it? How have you seen that you have performed a project that is similar and even competitive with other projects that you can see on the project wikis?
So if I may speak from
I apologize,
experience.
but we are gonna have to shut it here. It was a good question. I would have loved to hear the answer as well. But thanks, iGEM-Biotech-CCM. I'll let you lead, and I'll start letting the next team in. Thanks again for your time, and we hope you enjoyed your judging session. Talk to you soon.
Thanks a lot.
Thank you very much.
Thank you.
Hi there, team. Is there anyone else from your team around? On the waiting room?
Yes, Estefanía Gines.
Okay.
María del Carmen.
Okay. And is that it? Okay.
Yes.
Well, Thanks again for joining. This is your judging session. You have a maximum of up to 20 minutes. So if there's no one left to be let into the room, I'm gonna let your judges start asking you questions. So thanks for being here. We have Carlota and Mohammed as your judges in this room. So here we go.
Thank you so much. Thank you, Ricardo, again, and welcome for the team BioTec-CCM. Thank you so much for your project. We really enjoyed reading, going through it, and also we are excited to have you on this session. Also, please note that big part of this session is just to work on the improvements that could be done for your project rather than a simple assessment. We really enjoyed your project, and we wanted to start simply with your rationale that you used for selection of your targets for the diagnosis. Do you think that you could, generalize this approach into different diseases?
Okay, thank you so much for your question. Yes, like total switch, it's a very variable technology. The target of the pathogen or any organism of interest, it could be changed regarding of your necessities. So we choose two genes, one mitochondrial and one by the genome of the Babesia bovis, but it could be changed if it needed.
Thank you so much. So do you think that a mitochondrial gene could be more difficult, more challenging to detect in a biological sample? Do you think that you need to provide some enzymes to increase the yield of that target protein in your sample?
Yeah, it could be a little bit challenging to detect the mitochondrial gene because we are only trying to get all the ADN that we can get from the sample. But for that reason, we are we implement the RPA isothermic amplification. So it will allow us to, even if there is only copy of the gene, we could take it and amplify it.
Okay, so generally, do you think that there are some yield limitations on this diagnostic platform? I know that there is big literature on this, but do you think that you can overcome an obstacle of a limited yield or low yield in biological samples in general, not just for mitochondrial products?
Yeah, in general, we think that the first issue of the test would be to take the target, okay, the trigger that it's only 30 nucleotides, but that's the reason we implement the RPA amplification. So yeah, that would be the biggest issue.
Okay, thank you so much. So please, Dr. Carlota, I'm happy with these questions. If we have time, I can introduce some more points to discuss. Please go on if you would like. Okay, I think we can resume the discussion. So please, could you please tell me about the dynamic simulation that you performed for the thermodynamics of the toehold?
Excuse me, can you repeat it? I didn't hear you so well.
Sorry. So I was asking about your modeling. I know that you performed a very good job on this, the modeling of your toehold thermodynamics. So are you happy with this? Are you happy with the rate of the delta change in the general ensemble energy upon the interaction and with the free version of the toehold?
Yeah, so we analyze the free energy of the structures because we want to ensure that the complex is going to be formed. So for that, we make sure that the free energy of the complex is more negative than the unbound state. And that's help us to ensure that the complex is going to be formed.
Yes, great. Yeah, this is totally correct. You guys are all undergrads. Are you
Yes,
all
we are.
undergraduates?
Yes,
Okay.
we are undergrads.
So with different majors, or you major in bioengineering?
Mostly we are from bioengineering, but we also have integrants from another university and from mathematics, biology, and yeah.
Okay, great. So I would like also to know, how would you, I mean, consider the prototyping of your product? I know that you have provided some milestones on the PDF, but I would like to know from you, how competitive do you think your product would be in a market, rather national or international? Do you think that you can stand with price competition, with effectiveness competition?
Okay, yeah. So we are trying to build a kit, okay, like the kit with everything that you need to do the test. Talking about prices, we haven't done I can tell you a real price of the test, because first we have to standardize and optimize the use of some reactives that maybe are cost, but it's not it's not going to be a lot the amount of the reactive that we are going to use in each test. So this could be a process of a standardization, of optimization of those reactants, but we think that we could be very competitive because of our specificity and the lower cost, comparing it with the prices of the other tests like PCR and things like that.
Okay, so you have provided, clearly stated that that your product could reduce the waiting time for the diagnosis to be performed. Have you performed some quantification for that reduction using some estimation or something?
Yes, with modeling, we try to establish a time to the test. How long is it going to? How many time is going to take? And we are. It is according to the modeling, right? It's at approximately 3 hours, maybe less. It is going to depend of the in vitro testing.
Do you think that a farmer could perform it like without help, without scientific help, without some technical help?
No, this is not designed for a farmer because it's not ethical a farmer do the test, because he can hide the information, and we are trying to also establish a prevalence data here in Ecuador, because Babesia bovis is not a disease with numbers, with data. So because it's confused with all the hemotropics, so this is direct to maybe a veterinarianist, yeah, a veterinarianist or a lab technician. But the thing in this test is that you don't require a lot of or fancy equipment.
Okay, thank you so much. So I would just go a little back to some technical point, and then we can discuss about the impact of your project. So on the toehold designing, you know that these toeholds can actually have, there is no specific cut of like a binary, this will work as an on switch or an off switch. You know that it always have a likelihood of performing a specific function. So do you think that you have included an estimation for how the on and off activity for that? I'm sorry for the noise.
Yeah, I'm sorry. Can you repeat the last part? I didn't hear you.
I was just asking about the on and off activity of your toehold. Do you think that your simulations or your computational methods included that? that because this toehold might act as an off switch rather than an on switch, or vice versa.
Yeah, that is going to be part of our build and test proof of concept, because we need to get some data from the testing for lab testing for that. So we are proposing to take a relation between the on and off state and see how is it working. Michael wants to say something.
Thank you so much. I see also that you have included a tool that can actually estimate a probability of having this on and off activity. So this could be extremely helpful for you on this point, specific point.
Yes, we had used a rate of this K-on and K-off. This rate was obtained from software and proposed from another iGEM team, and so we introduced our sequence of our toggles, and it says this rate, and with this information we calculate an estimation of our own K-on and K-off values.
Okay, thank you so much. So do you think, going back to your bioentrepreneurial journey, do you think that you are ready to apply for some incubators or accelerators at this stage?
I'm sorry, I didn't understand you.
So regarding your bioentrepreneurial journey, do you think that you are ready to apply for startup accelerators?
Yes, for sure. We are pretty sure of our work. We did a lot of work, and we are confident, and this test is going to work. So we need to do a little of testing, of lab testing before everything. But I think it's going to work. It's going to be helpful for this, for the diagnostic of the disease.
Yes, I think that. Well, we think that our model will act really good in the camp, because this is an self-free system. So we did in our modeling, we did an deterministic modeling. So as it is an self-free model, we know that there is not too much interference in the between the species, but however, we know that we need to go to the lab and see what really happened.
That's great. But you have really a great computational design. So finally, I would like to hear from you on your journey this year on the Design League. You are from different majors. Have you enjoyed it? Do you think that your project now stands out for the other iGEM teams on the wikis that you have been browsing?
Yeah, I think we have learned a lot. It's been a long journey, full of a lot of the emotions. Those are good, those some are bad, but I think we're very happy. I'm now very proud of our team and our work.
Yes, thank you so much. We are also proud of you. Would you add something to this from another member?
Yes, I would like to add that we had a lot of help from another institutes, and also for our professors. They were really important in our journey, not just for direction, but for their help and support in this journey too. Also, we had help from another types of careers, not just biotechnology. We had a mechatronic student that was helping us developing our modeling. So we are really happy to have all of these relationships formed during this challenge.
So great. Actually, I thank you that you mentioned this point, because this is extremely helpful for a successful project. So I would like to ask you on the mechanical design of your test. do you think that you can put it on a microfluidic chip?
If I can put what? Can you repeat it? Sorry.
If you can provide your diagnostic design into a microfluidic chip that can render it. I mean, I mean, you need some microfluidic design and expert, but have you considered this for prototyping or for releasing your product?
I'm sorry, I didn't understand the question. I think it's my internet or maybe yours.
I think that I understand
We
a
don't
little
hear
bit.
you.
And we really what we propose is a paper test. So we think that it can. We try to do it easy to use. However, as we have said, we know that farmers can do this because it has other process. But in general, we were thinking to create a very easy to use test.
And something to add, I want to tell you something about the social impact. And of our test is in long term, the farmers could avoid the economic losses that they could have, even the disease could have some critical cases, and they could avoid this extra money for their work, like production of the meat or milk or daily products.
That's great. Thank you so much. Thank you so much. If we have final time, I would like to know what are your future plans for your project in the near future?
Okay, yeah, we have plans to try to test it and get all the information that is missed for us. We need to take some constants, some proof of concepts that we need to execute in order to improve all the work that we have already done, and also try to make some testing kits to start optimizing and standardizing the process to build the test.
I would like to add something else. We were planning to, as Stefa said, that we want to build kits for hemothropics in general, because our product could be applied to any of the homotropics just by changing one part. And we also want to standardize our process, because by doing that, we will diminish the cost, and that's our biggest challenge right now.
Thank you so much. So you are very, I mean, sure about the modularity of your diagnostic kit. So it can just be a plug and play sort of thing for adding a different target or something.
Yeah, that's the wonderful of the toggle switch. You just have to change the target and it's going to work. Yeah, obviously you have to make some work behind, but this is the future for testing.
Perfect.
Do you have,
sorry.
Yes, thank you so much.
Yeah, we're gonna have to cut it here. Sorry about that, Mohammed. But then again, thanks again, Team BioTec-CCM. We're really glad that you were able to be part of this judging session. And we're gonna take some 10 minutes break, and then we'll continue with the next team. Hope you enjoyed your judging session, and we'll be seeing you soon.
Okay, thank you.
Okay, Mohammed, are you there as well?
Hello, Ricardo. Yes, I am here. Yes, I am here. I'm ready.
Great. So starting now, you're going to have up to 20 minutes to go over some questions of your project. So this is Team PANA, and you are a team from Venezuela. So welcome, and let's get started.
Okay. Hi.
Hello, Timbana. Thank you so much. I would like to congratulate you on your project. Can you hear me well?
We hear some background.
Yes, I'm sorry for the background noise. I would like to congratulate you on your project. And I will start simply by discussing some points which we really think that it will just help improving your great project so far. So it's not just an assessment, it's a very exciting discussion that we would like to have with you, because we understand how much the effort that you have put, which is really clear from your work. So let's start with you, Amin, with the selection of the consensus sequence for your antigen of interest. Do you consider a specific, a specific cutoff for mutations to be included or variant frequencies from the alignment? I really like this approach because it could, consider some variants that are evolutionary competitive for the virus, like the variants that are evolving for SARS-CoV-2. But what are the cutoff that depends on? I would say that I would choose the residues that have 50% of the chance appearing in the alignment sequences.
Yeah, maybe Javier has heard the question.
Okay, give me a moment. I have a little issue to connect here.
Feel free.
Can you hear me? I can listen you clearly.
Yes, we can hear you.
Okay. We select a consensus sequence for, because we need a total mutation spike protein coronavirus. And this is a problem with this is approach that this is our approach to convergence evolution to total mutations on total sequence in spike protein. Different variants have different mutations on this sequence. Delta variant has mutation on amino acid 614, the G to G amino acid sequence, and this mutation have prevalence on the rest of variants. But we don't know how many mutations has these different variants on spike protein sequence. I don't know if you can understand me. Excuse me for my English.
Yes, no worries. That's great. I thank you so much. So I really, just as I aforementioned, that this approach is perfect if you would like to consider targeting the variants of concern for COVID. So there are different sequences arising in different regions with different mutations. So that's what I like this approach. So let me go to the second point, which focuses on your immune predictions. So you have included predictions for cell-mediated immunity. So a lot of tools for cell-mediated immunity, but actually your display approach is focused on antibody display, like the epitopes. You are trying to test for an antibody response, whether it's linear or conformational. So what do you think the value of the cell-mediated immunity tools like NetImageCell or these tools?
Okay, give me a moment. Judith, can you help me with that part? I can understand because.
Sí, básicamente está preguntando que tú hacen como una predicción y una inmunopredicción y por qué el proceso está testeando por los antibodies. Entonces, ¿cómo haces ¿cómo sabes que el proceso que estás utilizando es el correcto para predecir la eficiencia más o menos?
I don't have words to explain in English. Juliet has a translator for you.
Yes, feel free, no worries. No
Excuse
worries.
me.
No, feel free, no worries. I think Juliet will translate.
Yeah.
Ok, déjenme, voy a decirlo por 2-3 oraciones y Juliet, por favor, vas traduciendo conmigo, please. La predicción basada en células inmunes permite entender o predecir hasta un cierto punto cómo va a ser el comportamiento del antígeno, entre paréntesis, sustancia exógena.
Okay, so the prediction of the no, that was too long. ¿Puedes repetirlo más corto y lo voy a traducir?
Okay. La predicción basada en células inmunitarias.
Yeah, so the prediction based on the immune cells.
¿Cómo será el comportamiento de ese antígeno o sustancia desconocida para el cuerpo?
And predict how the behavior of the antibody is gonna occur. Yeah.
Una vez que haya sido ingresado y reconocido por el sistema inmune.
Once it has entered the body and has been recognized by the immune system.
Entonces, no solamente se considera la inmunidad basada en anticuerpos.
So it's not only considering the immunity based on antibodies,
sino también basada en células linfocitarias T.
But also based on the lymphocytes T cells. I'm not a biologist, just to clarify. So.
No worries, that's a great. So you are trying to include also the effect of T helper cells because it will have a role on the recognition and presentation for these antibodies to the T cells in which the antibodies will be produced. So you are using a key function of the cell-mediated immunity in order to boost the neutralizing response of the antibodies. That's really clear. Thank you so much. So let's move to another point, which is based on your predictions for allergenicity. So let me first understand, but after you select a consensus of epitopes, do you rely on a specific, just a consensus sequence that results from the alignment, or you try to make combinations of epitopes using linkers or something?
Okay, Juliet, ¿puedes traducirme esta parte, por favor?
Yeah, but I'm sorry, but I'm gonna need you to repeat the question shorter so that I can translate as I go.
Okay, sorry. So let me first understand the final consensus antigenic sequence. Is that resulting from the alignment or resulting from multiple epitopes resulting from the immune predictions linked together by linkers or in another form?
Que si la última predicción, la última consensus, la secuencia, que si viene de una predicción de...
Alineamiento múltiple, yo lo entendí.
Ajá, de alineamiento o si viene del... No sé, lo perdí, de una...
Ok, yo al medio lo entendí aquí, puedo responderle un poco. La secuencia consenso fue generada por el programa HALView.
Yeah, so the consensus sequence was generated by the HALView. Yeah.
Ok. Y se basa en la frecuencia de aminoácidos en cada secuencia FASTA que tenga la proteína.
So it's based on every amino acid that the sequence FASTA has.
Y a partir de la frecuencia de aminoácidos y según su proporción sobre el total de secuencias,
and according to the proportion of how many sequences and the total quantity of amino acids,
se determina cuál es el aminoácido con mayor prevalencia sobre el total de variantes que se están estudiando.
You determine which is the amino acid with the... ¿Puedes repetir? ¿Cuál es el aminoácido de?
Se determina cuál es el aminoácido prevaleciente según el total de frecuencias.
Prevalent, according to the amount of sequences.
Entonces, a partir de allí, se tiene un mapa general de cómo es la secuencia consenso.
So from that you have a general map of which is the sequence, the consensus sequence.
Para el total de proteínas que se tienen en ese alineamiento múltiple.
For the total of proteins that you have in this multiple alignment.
Okay, great. Thank you so much. So yeah, are you using a product, a consensus product of the alignment in which it will be the antigenic construct to be used? Okay, so let's move to just a point in the predictions on allergenicity. So allergenicity tools are usually trained on antigenic sequence, so sequences of antigens, not epitopes. So I just would like to check, have you checked the predictions of allergenicity based on the consensus sequence or epitopes, specific epitopes?
¿Podrías traducirme la última parte, Julia?
Que si has considerado ya esto en base a los aminoácidos o base a los epitopes, porque quería saber si has considerado una palabra que no entendí.
Yo puedo entender un poco por acá, pero es algo largo de explicar. Voy a ver si lo puedo decir un poquito en inglés y si me pierdo te digo por aquí. Or non-allergenic and antigenic, or non-allergenic and antigen epitopes has created for immunoinformatic analysis from IEDB.org and Denmark Technical University. These peptides, we analyzed each one on iGEM version 2 version, approximately. And we determined which has non-allergenic potential, and we create a library with H1 products. Then this is most particular for not total consensus sequence. Aquí ya me perdí un momentico, Juliet, sino también para cada péptido corto.
But also for every short peptide.
Okay, I see. Yes, thank you. It's clear now. I think it would be, more straightforward. Fit on training to use the consensus sequence, but of course using the peptides, yes, that's fine. But it's more, these tools are more fit on antigenic sequences, so I think it's more valid to use just the consensus sequence.
So que él cree que utilizar la secuencia de consenso directo es como más directo.
Ya, perdón, ¿lo puedes repetir? Se me blagueó por acá.
Que él piensa que es más directo utilizar la secuencia de consenso.
Puede ser, pero hay que tomar en cuenta de que no se toma toda la proteína en sí.
So it could be, but you have to take into account that you're not taking the whole protein itself.
Sino los péptidos cortos con los cuales está formada esa biblioteca que tiene iGEM.
So, but only the short peptides that makes up the library that has iGEM.
Porque si probamos la proteína total, nos va a decir segmentos que reconoce, pero se pierde información.
Because if you use a total protein, then you're going to see segments that are recognized, but then you lose information.
Yes,
y estos péptidos son los que realmente permiten discernir cuáles son los útiles.
Those peptides are the ones that actually allow you to differentiate.
Yes. Thank you. I like the enthusiasm that you are guys having for your project. It's really great. So it's just a point of how these tools for allergenicity prediction have been trained. They are trained on large sequences. So if you provide small sequences, they just will not be so confident about prediction. So I really liked also the way that you used the M13 phage proteins. So are you intending to use this as a vaccination platform or as a peptide display platform to detect antibodies?
Que si lo estás utilizando como una plataforma de vacunación o como una plataforma para detectar los anticuerpos.
Plataforma de vacunación totalmente.
Vaccination platform, yeah.
Great, very, very well said. So do you think that this supplementary action of having this as an antibody detection platform, like for autoimmune diseases or something, do you think you are intending to add it as an auxiliary function?
¿Piensas añadir como una función auxiliar esta como plataforma de detectar detectar los anticuerpos para otras enfermedades inmunoatacantes?
Puede utilizarse, de hecho, y serviría, por ejemplo, con la diabetes de tipo 2 en tal caso.
It could be used, and it could also work for example, diabetes type 2 diabetes.
Okay. Yes, please go on. Okay, thank you so much. So this has been clear for me. So I would like to also to hear from you on your experience on the iGEM Design League this year. You guys are from one major, from different major, and how do you see your progress according to the, I see that you have achieved a lot of milestones. So have you enjoyed it? Have it been stressful?
¿Cómo ha sido la experiencia durante iGEM? Que quieres saber un poco de parte de nosotros, que si somos de un mismo programa, diferentes programas, cómo nos ha parecido el programa y si lo has disfrutado y aprendido y todo.
All members, we are from different universities, countries. Mentor live in Netherlands, and the rest of team group live in Venezuela. 5 members are from Universidad Simón Bolívar, Universidad Central de Venezuela, Universidad de los Andes, and this experience integrate all knowledge for biology, synthetic biology, data science, and convergence in bioreactor design and engineering support from our mentor. Esta experiencia, ahora sí puedes traducir por mí, por favor. Esta experiencia ha sido de lo más diversa en el aspecto que podemos integrar distintos conocimientos a la vez.
So yeah, for him the experience has been great because you can integrate a lot of knowledge and a lot of different fields at the same time into one project.
Y que también desde la adversidad hemos podido estar en contactos todos y tener un objetivo común de trabajo.
Yeah, and of course it's difficult due to Venezuela's situation to keep track of everything and the communication, but this project has like unified the goal into one project and one idea.
So do you think that this project, during your human practices, do you think that you are going to be like a junior center for synthetic biology in your nation? Do you have this reflected in your community? Do you think that you could start more projects?
¿Piensas que puede ser como el inicio de empezar a más proyectos, de ser como un centro de biología sintética, una organización y seguir adelante con esto en la comunidad y algo así?
Sí, a futuro podría establecerse un centro de biología sintética a pequeña escala desde centros de organización civil. Estamos planteándonos eso como equipo.
Yeah, so as a team, they see as an option to become like a small mini center of synthetic biology in the area, in the country.
Y que nos permita actuar de forma descentralizada.
And it allows you to act not under one institution, but as an unified effort of the nation and everyone that wants to focus on synthetic biology.
Yes, that's pretty impressive. So finally, do you think that your vaccination platform could be a candidate for a bioentrepreneurial journey? Do you think that you can start a startup journey here in iGEM?
Si crees que este proyecto puede ser como un inicio de un startup, como que para la vacuna, para empezar una especie de proyecto de entrepreneur.
Sí, lo vemos de esa forma. En el equipo también estamos planteándonos saltar a esta segunda etapa, por así decirlo.
Yeah, so they see it as a possibility. They've actually discussed within the team, the possibilities of taking it the step further.
Y así arrancar no solamente con este proyecto, sino con variantes del mismo.
And not only start with this project, but also with variants of the project, as we were discussing the auxiliary platforms that you can create with the same approach.
Great, thank you so much. So I'm so happy to have this exciting session with you, and I wish you the best of luck in your project and your career. Thank you so much.
Yeah.
Thanks
for the opportunity.
So we're gonna have to leave it here. We're gonna allow the next team to join us, but first, again, I'd like to thank you both, Mohammed as a judge, and also you team from Venezuela for being the first team from Venezuela to ever participate in iGEM. So congratulations, and we hope you enjoyed your judging session. We look forward to talking with you soon. Thanks again.
Thank you.
Thank you.
Hi team, I'm allowing all of those who have registered as a team member. Are you expecting someone else?
Wait. I think we're getting some kind of like it's repeating itself, your voice again.
I don't know if someone has the live stream on or something. Let me check. Are you waiting for more team members? And if so, who are they?
We're waiting for Mateos. Kyle.
Okay.
I think we have a lot more actually. Joanna.
Joanna.
They have changed their names before. Danielle. Wait. Beatriz. I think Isabella.
I have Fernando, a Daniel, and a Gabriel.
Daniel, Gabriel.
Mm-hmm. So I think everyone's in.
Is it the voice? Is it repeating for you guys too? It's really bad for me.
Are you watching the YouTube transmission too?
Wait, can you say that again?
Are you watching the YouTube transmission?
Okay, I got it. Thanks.
Okay, so starting now, you are gonna have up to 20 minutes. So we have here our judge who has questions from the other judges as well. And welcome. Feel free to start with the questions, and we're glad to have you here on your judging session.
Hello, Team Ararinhas. Am I speaking it correctly or?
It's Ararinhas.
Okay, I think that would be difficult for me, but thank you so much for your project. I really enjoyed reading your files, going through the juggle. Thank you for your effort, the dedication that you have put, also the scientific effort. I really like it so much. It's because it's one of the finest therapeutic projects that we can see. I would like to start discussing with you some points, which be sure that they are just part of it is about improving the project rather than a simple assessment. So let's start by the rationale for your therapeutic approach. So usually when we deal with brain tumors, we are hindered by our delivery method because actually we have the blood-brain barrier. So how do you think your approach could, overcome this, or how this could be tackled by your delivery method?
Vocês entenderam? Hi, I'm here to help.
The voice stopped a little in the end of the sentence.
He wants to know, I'm sorry. Ele quer saber se como que vocês superaram. I'm sorry, Mohammed. You want to
Yes.
know how they overcome
The blood-brain barrier. The blood-brain barrier.
Barreira hematocefálica.
Yeah. Como vocês conseguiram superar os problemas de acessar a barreira encefálica?
Okay, we got a meeting with a researcher from our university, and he gave us the idea to guide the salmonella for our treatment by an artery from the tumor by catheterism. So we would use it to make it arrive directly on cancer.
Okay, great. So yes, actually, this is well said, because you have different approaches. You can provide it intravenous, but this approach is actually not so, doesn't have a good potential for brain tumors like glioblastoma. But actually, you can have a catheterization, that a procedure that is done clinically by introducing some drug or some therapeutic component into an artery. Usually, it's minimally invasive procedure. So yes, you are right. So let's move to another interesting point about your approach. Do you think that delivering your engineered salmonella could infect healthy cells of brain?
We really trust it won't, because salmonella has a natural tropism to tumoral regions, and we will improve it by adding the peptide RGD. So we will test in laboratory, but we do trust it won't help. It won't happen. The salmonella won't infect healthy cells.
Can I add something to that?
Yes, please.
Yeah, so if it does get to any kind of normal cells, actually our type of, our strain that we're using, it infects cancer cells over like 1,000 times over normal cells. So this chance of infecting normal cells, it would be very minimum. And also our strain depends on purine, which is found in tumor regions. So if they're not around tumor regions, more likely they will die.
So do you think you will benefit from adding a logical module that, decide upon cellular interaction or cellular recognition? So it works with these cellular receptors and which is specific to the cancer cells, the glioblastoma.
So you're asking us if we're confident that we're gonna engineer it to be more specific to the glioblastoma receptor?
Yes, do you think that you would consider this approach?
Yes, we actually consider it, and another researcher at our university who's specialist in glioblastoma, he said that there's a specific type of receptor that it's overexpressed in glioblastoma cells, which is a kind of glutamate transporter. So if we can find a way to actually link to that, it would be very specific to glioblastoma.
Great. So you can actually find a lot of synthetic biology approaches to cell specificity. Like we actually recently developed technique for gene delivery is called SEND, which depends on degalylated proteins, which you can, incorporate with some fusion proteins that is specific to cellular receptors. So you can try this, you can try different methods, but I actually like your approach and your confidence on how it works. So let's move to discuss another point, which is the apoptotic effect of the caspase. Have you tried some network modeling? Do you know the signaling pathway that will be, exerted upon the caspase expression? Do you think it will be enough, or it would need another apoptotic signaling?
Well, the caspase acts in the end of the apoptotic process, so we think it would be enough to make the cancer enter the the apoptosis. But we will test it too. We plan to go to lab and mix the culture with salmonella and glioblastoma cells to make it sure. But we think it will be enough.
Thank you so much. Really well said. And I actually like as well that you are depending on a specific level of the signaling pathway that is terminally effective. So, but let me also discuss with you, have you used some modeling that made you sure about the strong apoptotic effect upon the caspase expression by your system?
Can you repeat that? I'm not sure I understand the question.
I mean, if your modeling has provided you some insights on the effectiveness of the caspase to induce the apoptotic effect. Or in general, do you think that your modeling have helped in shaping this design?
I mean, we didn't do any actual modeling on the kinetics of the enzyme and how it will work in the whole cell, but we're hopeful that because it is the same protein as it is in the human cell, it will most likely work.
Okay, so you are sure that you will achieve an expression that is terminally effective for that function?
Yeah.
Okay, great. Really nice. So I think this is really impressive. I don't have much on the to question your engineering. I think it's really helpful. I would just ask you a general question. Do you think that your approach could be effective, I mean, modular for other cancer types? Like if you want to treat other solid tumor in different region or a blood tumor that is, I mean, infiltrating the whole circulatory system?
Yeah, I think it would be helpful to treat another types of cancer because all of them or almost all of them have some particularities in common to cancer regions. So the salmonella would be guided to these regions in the same way.
And also as to the caspase 3, I mean, that could be modular, right? You really, I mean, you're giving back the capacity for the cells to undergo apoptosis. So that's always good for cancer cells. But also you could, I mean, you would have to dig into the balance between anti-apoptotic proteins and pro-apoptotic, because in our case, there's a high level of BCL2, which is an anti-apoptotic, and low levels of caspase 3. So you would really have to get into that first.
Really nice. Thank you so much on this. So, do you have some considerations for the toxicities that might come alongside with salmonella infection? I mean, the tumor microenvironment usually have some key interactions with immune cells, with healthy cells. The cell-to-cell interaction actually provides some minimal, I mean, some minimal collateral damage, if I would say. Do you fear, I mean, salmonella could release some toxicities into the healthy cells in the environment?
Right. Actually, our strain is free of toxins that would be very harmful to the human, I mean, the host and the brain in general, but also because the glioblastoma is such a unique type of cancer that actually modulates the environment of the brain and microglia, that could benefit. I mean, if we lower the levels of glioblastoma, then the microglia could actually properly work.
Okay, really nice. Thank you so much. So I would like also to hear from you on your human practices. How did it shape your project, and how do you think your project could affect the community, especially the healthcare community?
Yeah, I think the glioblastoma is a quite aggressive type of cancer. So this way we can treat the tumor with a less aggressive way that the traditional surgeries that it has, and chemotherapy and radiotherapy. So it's less invasive and acts only on the area of tumor. So it's less aggressive. I think it's the correct word. So
yes, really correct. So do you have plans to provide some approval applications and the FDA associations in your nation or in different nations for your as a therapeutic approach? Yeah, do you want to enter the clinical trials?
I mean, the professor we talked to, he's a neurologist at the university hospital. So he was very, very excited to hear about our project. He said that he has patients with limited life expectancy. So that would be really cool to introduce as soon as possible. He's really excited about it. So definitely, if we have more support, we'll definitely try to go into it.
Yeah, and he's really supporting us, and he said that as soon as the university allow us to go to lab and we test it more, we could work together to develop the treatment.
Thank you so much. This is well said. So
you're muted.
Oh, sorry, sorry. I would like to know finally from you about your backgrounds. What are your majors? Are you in the same major? And how this shaped your project? And how do you would like to maintain your project in the near future, if you would like?
Right, we're students from biomedicine and biology, and in biology, it can be divided into bachelor's and the teaching aspect of it. So we have like all three kinds of here. We are really excited to build this health project because most people here are really excited about this field in biology. So I think we're gonna do an interesting job for the next months.
It's so interesting. So how do you describe your journey in iGEM? I think it's your first time to join an iGEM project.
To most of the group, yes. Just Luisi, our leader, was from another team last year, but it's completely excited and it's hard, but it's worth it because it's really nice. All of us learned a lot during this year, and all of this started during our classes with our teacher, and we hope to keep learning that way to develop more stuff.
So are you happy with your project at the moment in comparison to other wikis that you have browsed before?
Yeah, completely happy.
We are happy with you too, and with your project is really impressive. So thank you so much. It was really exciting to meet you and know about your project. I would like to congratulate you again, and really excited to see you like in a bioentrepreneur journey, developing your therapeutics into the next level. Thank you so much.
Thank you.
Thank you.
Thank you.
So, yeah, so congratulations. You made it to this point. You are the last team for today. Congratulations, Team Ararinhas. Thanks again, Mohammed, for your participation here as a judge. Yeah, everything that you have done throughout the season has led you here, and we're very proud of what you have accomplished. So thanks again for your participation. We hope you enjoyed your judging session, and we hope to see you soon at the award ceremony. Thanks again, team. Have a nice day.
Yeah, thank you.